talk about comparison of volumetric registration algorithms for tensor-based morphometry. This is a co-joint work between myself, Natasha Lepore, and Joshi, Carolyn Brun, and Paul Thompson at UCLA, University of Southern California, UPenn. So the main motivation for this study was to find the best registration method in order to do tensor-based morphometry. For those who are not related with the term tensor-based morphometry, is a way to analyze, to compare populations and get the differences in anatomy between these two populations, let's say disease group and control group. And in a better way to explain it, if we have group 1 and group 2, we have to align them to a common space, to a common coordinate system, which is usually done by a registration method to a target. And after we have the deformation field for this registration, we have the Jacobians, and most common methods in tensor-based morphometry use the determinant of this Jacobian to get the comparison corresponding compared to the target in order to see if the tissue at that particular point got expanded or got reduced, as we can see in the figure on the right, where the blue areas are the ones that expanded and the red areas are the ones that got reduced. So we compared 3 registration methods. The first of them is a 3-dimensional whole volume fluid registration in a Riemannian framework, which has been already published by Brun et al. We also used a commonly used non-rigid registration method called diffeomorphic demons. And another motivation was to see if combined surface volumetric registration methods, already published by Yoshi, could do better into finding fine deformation field variation across population. So why this motivation? Because it is well known that 3-dimensional whole volume registrations do good alignment of subcortical structures, whereas so they are not very accurate into aligning cortical structures, as you can see in the figure in the first row, which is a typical fluid alignment registrations where the internal structures are well aligned and the cortical structures are not so well aligned. But the problem is with these cortical surface-based registrations, where you initially segment the surface and then using FreeSurfer or other tools you can align these cortices, you're not taking into account the internal structures. So how did we compare the registration methods? As our main motivation is to find a good registration method that is able to find the complexity of the deformation fields, the finest differences between two populations, between the target image and the template image. And we used a twin population of 340 subjects, and we used a statistical measure called intraclass correlation, which is a method that is able to see how correlated a couple of twins is in relation compared to unrelated subjects. So what the registration method is intended to be able to do is to pick the similarities between twins related to unrelated subjects. So this is work already presented in ISBI 3 years ago, 2008, and this is a cool result that we can find after we have a very good registration method. What the authors did here is they took 10 twin pairs and they registered each twin to the other twin pair and to the other unrelated subjects. That's what you have on the x-axis. And the y-axis you have a deformation field measure based on the deformation field. And what we can see here, the blue dots in each column, in each x, is the other twin, the twin pair of the twin and the other balls are the unrelated subjects. So what we can see here is that actually twins are more closer, twins' brains are closer to each other than to unrelated subjects. And this is also supported by previous research that the brain anatomy is under strong genetic control. So as again, the registration methods that we use, and I'm going to shortly explain each of the registration methods that we use, is a three-dimensional volumetric fluid registration, diffeomorphic demons, and the combined surface volumetric registration. The fluid registration that we used is a common fluid registration, where we model each voxel in the volume as a viscous fluid. We used, in this particular fluid registration, the cost function is the sum of squared intensity differences, and the regularizer is a log Euclidean variation in our Riemannian metric. And this particular regularizer ensures large deformations, and it's also diffeomorphic. And this is a typical picture where it shows the ball that in each time steps deforms into the seed. This is a previous method already published by Brun. The Demons registration is a very widely used non-rigid image registration, which is a non-parametric registration modeled as a diffusion process, and it's inspired in optical flow equations. And in each deformation step, it computes the forces of the deformation and regularizes by a simple Gaussian smoothing. And the particular version that we use here is a diffeomorphic version. And the combined surface volume registration, we initially segmented the cortices, and we did an accurate local alignment of these cortices using FreeSurfer. And then with the deformation fields of the cortices, we extrapolated them to the volume via a harmonic mapping. And once this was done, we fine-tuned the internal structures with a volumetric intensity registration, which in this case was an elastic registration, published also by Yoshi. These are the first results. Here we took all the registrations of the twins to the template, and plus the siblings. That's why you see n of 500. And we took all the registered images, and we calculated the mean. And what we can see here on the right, corresponding to results of the fluid registration, is that the internal structures are very well aligned, whereas towards the cortex, we can see that the cortex smoothes a bit, and the cortex is not well defined. For the Demons registrations, we got also a good internal structure alignment, and towards the cortex, it got a little bit better than the fluid registration, but where we could see good internal structure and cortical alignment was for the surface volume registration, once we did this average across all the registered images. And these are the interclass correlation results. As I said, the interclass correlation is a statistical analysis where we can see how correlated a couple of twins is related to, compared to unrelated subjects. So what we see here is areas of significance, which are red, and blue areas that are not significant. So the areas of significance are showing us the similar area, the most similar areas between the twins compared to the unrelated subjects. So what we can see here for the fluid registration is that the internal structures were more similar across identical twins. I forgot to say, the superior row is showing you the identical twins comparisons, and the inferior row is showing you the fraternal twins. MZ and DiZ stand for monozygotic and dizygotic twins. And as expected, we saw more correlated significant areas, p-values, for the monozygotic, the identical twins, compared to the fraternal twins. For the Demons registration, we got a similar result, a bit more smoother, and we think that's because of the regularizer that it's used, and it's also centered into the internal structure with certain cortical structures also included in the significant areas. And for the cortical fluid registrations, is where we saw the more results, including internal structures as well as cortical structures in the temporal lobe, intertemporal lobes, and also in some areas of the occipital region. And again, it was consistent with what we were thinking of, that if these areas would be more significant areas compared to fraternal twins, which share obviously less genetic, have less less genetic information between fraternal twins and identical twins. And yeah, this is our concluding slide, which shows that we already have published in ISBI'11 another paper where we also used a combined volume surface registration, but instead of using an elastic registration for the latest step, we used fluid registration. And the results of the comparison of these two methods, both combined fluid and combined elastic registration, will be published in the next weeks. Thank you, and if you have any questions.
We have plenty of time for questions. Are there any questions? Let me start.
So basically, you apply 3 different legislation algorithms. But from what I understand, in each legislation algorithm, you have the formation of the smoothness constraints. I think, in these 3 different methods, you may use a different amount of smoothness on the deformation field. So in that way, you identify difference will be very, very, different across the different three methods. Just think, think about, for example, in the legislation algorithm, if you put a lot of smoothness on the deformation field, that means, similarly, like you put a large, smoothing kernels, when you compare the two protocol images, right? So in that way, it's very difficult, for you to compare the 3 different methods.
Yes, you're right, and we're working towards this direction too, in order to set the regularizer and the cost function to be the same across the registration methods, in order to have to do better comparisons between the registration methods, because that's true. That's what we actually saw between the Demons registration and the fluid registration, where they used completely different smoothing and regularizing approaches. And yeah, it's definitely, you're right, and we're working towards that.
Okay. The second question is, in the beginning you mentioned like genetic identical twins, they are more similar compared to other subjects. So for the registration algorithm, you use basically you just each subject in a twin pair, independent to the atlas. So if you assume 2 subjects in the twin, they are more similar. So why not just adjust them together? For example, adjust them to their mean image space. Yeah. Then you adjust their mean to the atlas. And in that way, they are going to adjust, jointly to the atlas space. So you can avoid a lot of, noise, right? Probably in the original space, they are very similar. But, after you apply your pairwise, independent registration algorithm, they can become very different in the atlas space.
Yeah. I probably forgot to tell, but we didn't use an atlas as a template. We used a completely unrelated subject as a template. Yeah, but as we don't include that subject into the statistical analysis, what we actually want to find is how well the registration is doing into registering one subject with a completely unrelated subject to the study that's included into the statistical analysis. So we didn't use the atlas.
In this case, you make a registration algorithm much difficult. Why you need to, select the image very different to the twin. Like, they just, basically what you need to compare is, the twin difference, right? So,
Yeah.
so they should adjust together to any, atlas or any individual subject you select as a common space.
Yeah, but then what you could think after that is if we register each twin to each other, as the brains are more similar, then we're kind of biasing our comparison, because if the brains are more similar, obviously the registration might be better as compared to not so similar brains. So that's what we decided to register into a completely different subject, not included in the statistical analysis.
I kind of missed the slides. You promised us to show which registration separates the groups better. You have shown us a kind of...
No, this is the motivation of finding a registration method that actually is capable of picking the smallest differences in the deformation fields between in the registration.
So my simple question is, which one performs now better? You have shown spatially they pick up different things, but can you tell?
For us, is the combined fluid registration, because it's capable of finding areas in the internal volumes and also areas in the cortical rim that are more correlated.
Important.
Yeah, that's very important, because what we want to do is compare populations, and we want to find in the internal structures and in the cortex which areas are different across populations. And as you can see, as you saw, for three-dimensional volumetric registrations, they do better for internal structures than for the cortex. So we want to see both cortical and internal structures.
And this one slide where you had like all these subjects, how well they correlate against each other, just having one value for that wasn't that meaningful, or that wasn't really the result you wanted. I mean,
yeah, the results were the P maps and the mean of all, because the results were the P maps where we saw that the best the significance areas were in the internal structures as well as in the cortex, because we want to see differences in all areas of the brain across populations.
Okay, thank you.
Are there any questions? If not, let me conclude this session, and let's thank all the speakers for excellent talks and the audience.